This is a topic that is certainly new to me. I think probably new to many people. And I think in addition, I think Nikita is going to talk about tissue engineering also. So I think it's a novel topic, and I am eager to learn a lot from what Nikita is going to talk about. So let us start the class without much delay. Over to you, Nikita.
Thank you, sir. Good evening, everyone. So today's class is going to be about translational medicine. It's relatively a new branch of medicine. We'll first briefly go through about the translational medicine, then few points about tissue engineering. By definition, translational medicine means uh, within biomedical and public health research, it aims to improve the health of individuals and the community by translating the findings into diagnostic tools, medicines, medical devices, procedures, policies, and education. Basically, it's like converting whatever the newer advances and newer findings in the technological field, biotechnological field, and bringing them to the hospital setup. So it is often described as from bench to bedside. That is from the laboratory bench to the patient bedside. So, what the main concept of translational medicine is that it, as mentioned, the promising laboratory discoveries which are already in the which are already established, they will get converted into clinical applications, and these are attempts to answer the clinical questions with the use of all the uh, recent advancements, new predictions, new for the prevention, diagnosis, and treatment of the diseases. In other words, it transforms the basic research into the practical applications. Going back to the history, the first time the word is coined in 2003. The Institute of Medical Clinical Research at roundtable described the current terminology and the model of translational research as a two-phase process. The first phase being from basic science to clinical science, second phase being clinical science to public health impact. Later, in 2008, in a more detailed description of three phase was done. The first phase being from the biomedical science to clinical efficacy or clinical knowledge. From the knowledge to the uh, healthcare is the second, and to the healthcare and to the basic population would be the third. Currently, the 4T model is being in the application. That is the first phase being from basic scientific discovery to the potential clinical application. The second phase being the potential turning into an evidence-based guideline. The third phase being those guidelines coming into the clinical care and intervention. That is the applied knowledge. Phase 4 is the health of the community or the population. That is overall public health knowledge. So, what is the need for the translational medicine? With the current recent advances, with the enormous information what we have with the biotechnological advances, it is more important that we apply them into the current medical practice. Without that, the whatever the research, whatever the newer technologies which have been found will be of less effective. So there is a need for the continuous advances in the practical application, and that also reflects the gross national product in most countries. Translational medicine is usually a bidirectional concept. It which means that whatever the applications we are doing in the medical science, we need a feedback back to the laboratories for an improved and effective functioning. So this will encourage the flow of information from the laboratory to clinic, and in the same way, it should be encouraged from the clinic back to the laboratory. It means so the main aim is to increase the efficiency by which new therapeutic strategies develop through basic research and are tested clinically. Bedside to bench factors, which provide feedback about the application of the new treatments and how they can be improved. This is about the translational medicine. The main
Wait,
concept
wait.
behind translational medicine is sir.
So, Praveen, what are the three pillars of this particular concept? They describe three pillars.
Uh, Sir, it's a lab to with transformation into the
No.
basic
You
applications,
either
sir.
know it or you don't know it. Just don't waffle. Vinod? Nitya?
Sir, here the three pillars is first the clinical research, then the second is the evidence converting into an clinical practice, then it goes into the public health impact, sir.
Yeah, but you're describing the concept, but you need to tell me the three pillars.
Bed to bedside to community, and from the community to the again to the lab.
Correct. So it is the bench side, bedside, and community. They are the three pillars. So you can put the arrow. I think she said it's a bidirectional concept. It's probably a tridirectional concept because it goes from all of these. So, can you give me is Sania there? Hello?
No, sir.
Sania
No,
is
sir.
not there. Okay. Okay. Nitya, can you
Yes,
give
sir.
me an example of recent times where translational medicine has really worked?
Mm.
Maybe COVID-19, sir.
COVID vaccine.
Correct. How?
This mRNA vaccine production.
Correct. I think you hit the nail on the head. Basically, I don't know whether they used artificial intelligence, but they certainly collated all the information about how vaccines are produced and what is the principle behind vaccine production. And quickly, because it was getting to such proportions where it was estimated that millions, if not billions, would die if something quick was not done. So they collated the information from vaccine production of the previous years, and I think they genetically engineered it to so that they could ramp up the production of the vaccines with great speed. And actually, I think it helped to save the humanity. I think otherwise the death rates would have been probably a lot more, though people who actually didn't take the vaccines may disagree. But by and large, I think the concept worked and it made a huge difference in containing the pandemic. So I think we'll ask Lakshman sir's opinion, and then we will discuss what are the advantages of this particular translational medicine.
Very true. This is a very broad umbrella. A lot of things come under translational medicine, but one of the main activities is the use of large data, bioinformatics, and information about proteins, what they call proteomics, to develop new drugs and vaccines and the like. And as you rightly said, the process becomes much faster, more efficient, and more reliable and less expensive. So new drug development is one of the best examples of translational medicine.
Yes, sir. I think is it true, sir, that they can probably cut short the level 1, level 2 to the trials?
Very true. It is actually happening. So they are
Happening.
not going by the traditional model of drug development at all now.
Yeah,
So
I
that
think
is
the
what
other
happened
thing
in
is
Covaxin, this thing also, ICMR and that National Institute of Virology. They skipped this thing and they went in straight for trials on monkeys, on rare monkeys, which they had to go deep into forest and collect. So that is what happened in Covaxin, how the India
yeah.
produced Covaxin.
Correct. I think the what they say is that, instead of, for example, one size fits all, the chemotherapy for cancers used to be that you start everybody on the same chemotherapy. Now what has happened is, because of the translational medicine, you identify the particular tumor, and you identify the biocharacteristics of the tumor, and then maybe focus on what medicine will actually work on this particular person. So the AI has, the data, as sir said, data mining has gone to such an extent that they can now pinpoint which particular, chemotherapeutic drug will, work on which particular person. Of course, there can be minor mistakes, of course, but by and large, more focused and more targeted therapy is possible by this method. I think that is the major advantage, so that you can reduce the side effects and also maybe increase the potency of the particular chemotherapeutic regime as far as that particular person is concerned. And the other thing is also dosimetry also will be impacted by this, because they can understand the tumor characteristic, and then monitor and fine tune the dose of the chemotherapy. And also other thing is, in terms of the immune therapy also, immune therapy is also been made very potent, as far as one of the therapeutic options against malignancy is concerned. And this will actually go further forward by using the translational medicine. Sir?
Totally agree. I presume Nikita is covering all the aspects that this phenomenon covers. You're talking about various applications, Nikita, going forward?
No, sir. Actually, this is just an introduction to the tissue engineering.
I
Uh,
see. Okay.
so
Okay. I mean,
this,
you give your presentation, and then, I think
Yeah.
it's important to understand what are the areas where translational medicine is being applied as of today. If there are something that I need to add, I will add in the end. Please go ahead. Yeah.
yes, sir.
The reason is that, you were there at the quiz. We had one section called cutting edge technology in
Yes,
the
sir.
quiz, and so I had done some reading up about all this recently. So let's see how much you cover, and if there are things that I think people need to know, I'll chip in. Go ahead. Yeah.
So the topic for the discussion is tissue engineering. There are three main components of the tissue engineering. This is called the triad of tissue engineering, that is cells, materials, and molecules. What are these three components are like for any strategy of tissue engineering, we need three types. One is the cell for the replacement or for the regeneration. Second thing is the positioning of the cell, like um, a vehicle to transmit into the body. And the third thing would be the inducing substance, that is the molecule. First, we're going to talk about cells. The usually any
Nikita,
process
what
of
is term? What is term? TERM.
sir, tissue engineering and regenerative medicine, sir.
Correct. I think that is again another buzzword which is
Yes,
now
sir.
being used very, very frequently. Okay.
Yes. The main importance of cell is that its ability and capability to regenerate and reproduce. So here the most important point would be the criteria for using what type of cells for the tissue engineering. First criteria is whether the cells are sourced from the patient or from the donor. Because if it is from the donor, there is a chance that there is going to be a reaction and it's going to affect the outcome. And the second criteria is using like for like kind of replacement. Like For a chondrocyte implantation, we need to use for cartilage implantation, we need to use a chondrocyte type of cell. And for the third criteria is whether to use committed cell types or unspecialized cell types. That is, should we use a differentiated cell or a stem cell? So depending upon the situation and depending upon the requirement, we can use different types of cells. Most commonly used cells in the tissue engineering are mesenchymal stem cells, hemopoietic stem cells, and neural stem cells. These are the examples of multipotent cells. These can differentiate into a small subset of cell lineages. And a common alternative for the pluripotency are the any cells from the three germ any cells which can generate into three germ layers. Those are called pluripotent. Recent advances is the generation of induced pluripotent stem cells. These can be generated by reprogramming the fibroblast. So this opens a wide and uh, extensive opportunity to utilize these induced pluripotent cells in various uh, treatment modalities. And in a controlled in vitro differentiation, we can differentiate them into a wide range of therapeutic cells. The most common application is the skin and the cartilage. So first, using stem cell therapy, skin or skin kind of derivative is generated in the lab and which can be used onto the wound site directly. And in cartilage, chondrocyte type of cells are introduced into the joint zones or the area where we require a cartilage, and it grows into it. Second component is material. It is also called scaffold. It's usually a three-dimensional structure which is interconnected and has a porous network. This usually supports the cellular growth and also proliferation and differentiation to form different types of tissues which we are requiring. Factors related to nutrient supply, such as the porosity of the material, the size and the pore structure, the volume, these are all the critical important factors for the growth and development of the cells within the material. Generally, tissue engineering scaffold should be biocompatible. That is, it should be non-immunogenic and non-toxic to the living cells. And second point would be it should be biodegradable and capable of being remodeled and tuned uh, as per the host tissue. And it should be porous. That will enhance or encourage the host tissue to grow into the material. And the fourth point would be the surface should be conductive or facilitate the cellular functions which are required. It should be mechanically stable for the surgical handling, and there should be an feasibility and ease to manufacture and to sterilize the material. These are the requirements. And the common materials which are being used right now are porous. These are the various forms like sponge, mesh, microfibers, and nanofibers. These help cell aggregation and infiltration and further guide for the differentiation. Other examples would be hydrogel scaffolds. These contain extracellular matrix so that the cell growth can also occur within the material. Natural materials are used in hydrogels, including decellularized extracellular material, collagen, chitosan, and hyaluronic acid. These has more biocompatibility and with no inflammatory effect and less cytotoxicity. The other example is fibrous scaffold. It can be classified as porous microfiber or nanofiber scaffold. These also offer micro environment which is favorable for cell attachment and survival. Microspheres are the small ball kind of or spear shaped material. These provide biomimetic structural support and help in biological regulation and controlled release of biomolecules or the cells which we want to deliver into the host tissue. Other types are the metal and composite. Composite is the combination of different kinds of scaffold materials. The third component is the molecule. Molecules are generally of two types. One is the signaling molecule, the other is a bioactive molecule. So we have acquired a cell, we have attached it to the material and delivered into the host. But to signal the cell or to activate it into what kind of output we want to get, we need a signaling molecule that is handled by the bioactive and signaling molecules. So these are generally the growth factors like proteins or polypeptides and cytokines, neurotransmitters, hormones. These activate the cells and help them in the differentiation into the required kind of cell. Risks and challenges involved in tissue engineering are tumorigenicity, because sometimes the signaling and biomolecules are not completely under the control of the host. It results in the tumorigenicity. If it is an allogenic cell, there is a chance of graft rejection and immune reactions. There is a chance of cell migration and growth in a different zone in the host tissue. Others are the neovascularization and ethics is the major concern with the stem cell and the tissue engineering techniques. Cost is a major concern. It's a high-end technologies require high amount of manufacturing units and relatively higher
Hello? Hello? Nikita? Hello?
Hello, sir.
You suddenly
Sir,
stopped.
can you
What
hear me,
happened?
sir?
Yeah.
Yeah, we
Now I
lost you
can
for
hear.
a bit. Go on. Yeah.
Sorry, sir. Higher cost involvement. Uh, few of the applications of tissue engineering are in skin. There are a lot of skin derivatives right now in the market. There are of different types which are depending upon the manufacturing, depending upon the application. There are natural skin derivatives and synthetic skin derivatives, cellular versus acellular, and depending upon the composition, they are epidermal, dermal, and combined. So each component has a different kinds of different degree of uh, fibroblast and keratinocytes. This is one of the example of bioengineered glove of human skin. It is created for grafting. So this is all the skin which is made from the tissue engineering. It's a 3D printed. Other application is in bladder replacements. A study by Atala et al. Of 2006 with 7 patients. They had used adult autologous urothelial and muscle cells grown in the culture. for generating an urinary bladder. It's a biodegradable bladder, and it's the scaffold which is used is collagen and composite of collagen and PGA. About 7 weeks after the biopsy, I mean post implantation after 7 weeks, the engineered bladder was used for reconstruction and implanted either with or without an wrap. They reported excellent results in the follow-up period of 22 to 61 months. Another study in 5 patients where they had used smooth muscle cells and epithelial cells obtained from bladder biopsies. They cultured it for 1 week to ensure migration and proliferation. And the scaffolds they used were polylactic acids. And they had also reported excellent functional flow rates at 36 and 72 months. Other application is one of vascular grafts. In a multi-centric study of 2009 with McAllister et al., they have used tissue engineered autologous vascular grafts for creating vascular access in 9 patients who needed AV fistulas. And they had shown 60% of patency rate, which is comparable to the natural AV fistulas. Here, the scaffold used was a stainless steel material. Sir, this is just an outlook onto the tissue engineering and regenerative medicine. The conference of 2025. I was reading about tissue engineering and came across this, sir. The topics they have mentioned or the talks for the next year, they give a completely new idea into what currently the recent advances are, like to the extent of the applications and everything. I just wanted to share with you people.
Okay.
Sir?
Finished, huh?
Yes, sir.
Right. Now, let us ask others. Praveen. Hello,
sir.
Praveen.
Yes, sir.
What other than what Nikita has said, what else have you learnt? What else have you read about term or tissue engineering?
Sir, regarding the methods of producing the scaffolds,
Okay, tell us.
sir, it has different methods of gas forming, carbon nanotube method, solvent casting and particulate leaching method, emulsification freeze drying method, thermal induced separation methods.
Okay, good. What else?
And along in these applications, it can used in various other processes, sir. In creating heart valves in coronary heart diseases, in aneurysm patients.
Correct. Can you replace the smooth muscle of the heart after a patient has had a myocardial infarction?
Yes, sir, they have done, sir, valve replacement and muscle replacement.
Okay. What else?
And artificial skin, artificial bladders, artificial pancreas also they have done recently, sir.
Okay. Nitya, if Ainaz and Samir has got anything to contribute, or any questions to Nikita about what she has said so far? Have you all understood what she said?
Yes, sir.
Any questions or any other additions to what she said?
No, sir. So like I had just read that there was these different like based on the cells there were different therapy like replacement therapies. Embryonic was different, pluripotent was different. So like how, in which condition are we using what, sir? That like that, sir. I had that doubt.
Okay, Nikita, do you want to clarify that?
Sir, it actually depends on what kind of regeneration we want to make, sir.
Carry on. Don't stop.
Uh,
Beyond that, you
I
don't know.
So I don't know the extensiveness of the each component separately, sir, but the overview was this, sir. So embryonic cells we usually use as the pluripotent for any kind of differentiation. But to get embryonic cells is the problematic thing. So currently the easy assess thing is the induced pluripotent cells. We take normal cells and we induce them and use for the differentiation, sir.
So what are they doing for, childhood diabetes? What are they using for childhood diabetes? They're using the cord blood, umbilical cord blood, and then they are trying to make the destruction of the beta cells of Langerhans less possible and also increase the production of insulin by the islet cells of Langerhans in kids who are prone to or who have already developed childhood diabetes. So that is one form of genetic engineering. Nitya,
Yes, sir.
anything to add to what Nikita said? Any clarifications, doubts?
Sir, I read about something like liver tissue engineering also, sir. Like there is some bioartificial liver devices that are being getting developed in a it's in the clinical trial phase, sir. So instead of the
Tell us
transplant
more. No point in just.
sir, like there are like molecular extracorporeal liver assisted devices, and there are some bio assist liver support systems also, sir. Like exactly what they are planning to do is they will do something like what exactly the liver does, the same function they are planning to do it with these assisted kind of devices, sir.
So that is now you what you mean to say is basically as kidney has
Yes,
a
dialysis
dialysis.
kind of
So this liver,
system.
this is so you take over the function of the liver till the liver recovers, especially in acute liver injuries, usually drug
Yes, sir.
induced or viral hepatitis induced or ischemic liver injuries, where there is only way to salvage that person is to do a transplant. So instead of that, you just use these devices, allow the normal liver and the liver of the person to recover because liver can recover. So allow it to recover while this particular thing works for them and does the function of the liver till such time that the normal liver or the innate liver recovers.
Yes, sir.
Okay, that is
Yes.
one good concept. We'll go to, I think Sir will probably be the last person because he'll probably add more to whatever we know. We'll ask Niranjan. Niranjan, what are your thoughts about what she has said? Any additions or?
Sir, no addition, sir. I had very limited knowledge till today about this, so have to look into it. And I think it was a quite novel subject.
Same here. I think I learned a lot, from whatever Nikita has said. I think exactly. I also knew very little of what she was going to talk about. And unfortunately, she only showed it yesterday. Didn't even give. Lakshman sir, I'm sure you have a lot more to add to
Oh, yeah. I
this.
think, okay, first of all, congratulations on choosing the subject. Whoever did it, I think needs to be congratulated because postgraduates and all of us, practicing surgeons, I think must think about these things. This is the cutting edge technology in our field. And, by the by the next 2 or 3 decades, a lot of current surgical conditions will probably become medical conditions, in the sense you won't use a knife to treat them. That's the way science is progressing. I have very little more to add about tissue engineering. I think if you know this much about the cells, the molecules and the scaffold and the areas where they're being developed, I think it's very good. I'm just curious, I don't know the answer. For the Nikitha, tell me if you know, for the tissue engineered bladder, what about the neurology part of it? How does it feel it? How does it sense the feeling and things like that?
Sir, Nikitha, do you know? Otherwise, I
You know?
will tell the answer.
Yeah,
I
please.
don't know. It is just a guess, but
I
I'll
don't
tell
know,
the
sir.
answer. Hello?
Yeah, go on, please. If you know the
Sir.
answer, please tell me.
Sir, the thing is, there is what is now called the signaling molecules,
Yeah,
which apparently will, signal as
Yeah.
to how the bladder should function, and then I think it regenerates the pluripotent cells, will
Okay.
regenerate into the nerve fibers that supply the bladder,
yeah,
and I think that is the way they are going for even after patients have suffered strokes,
right.
ischemic strokes. I was wondering,
Yeah,
you can inject, stem cells, pluripotent cells into the area, but then who will tell those cells as to
Yeah.
what they should do? Because as you know, every neuron has a specialized function. So this signaling, I think the signaling may be at a very, very infant stage. It's a very, nascent stage at the moment.
right.
But I think signaling is probably going to become more stronger, and
Okay.
then ultimately it will signal what cell has to do what function.
Yeah.
And then that cell will start doing that function, apparently.
Fine. Yeah, stands to reason. There is a lot more. In fact, I think we should have it in another class regarding the translational medicine.
Yes, sir.
Basically, Nikitha has introduced only the terminology. What are the areas where it works? What is the current development? What is being done? It's a very, very wide topic. And honestly, I think we should have another class, even though I can, in 5 minutes talk about it. Before I close, maybe I should just tell you, translational medicine is, as Nikitha said, bringing the research to the bedside, and the reverse is true. Bring the bedside problems to the research labs, but it is a lot more than that. It involves a lot of ethics, it involves government policies, it involves communication, it involves information technology, computers, and things like that. And so now all the governments and other private research funding agencies are forming groups who will interact with each other to take this forward. And the topics that are covered are, enormous. It starts from cancer to chronic diseases to infective diseases. We looked at the example of infections in the COVID that we mentioned. Similarly, in cancer, they are focusing on early diagnosis through biomarkers and better treatment through targeted therapy. In chronic diseases, they are developing computational models which will help prevent heart disease, which will help prevent cerebrovascular diseases, strokes, and the like. And the whole thing can be considered under 3 catchwords. One of them is genomics, the other one is proteomics, and a new term that I learned very recently is metabolomics. Okay. Genomics is a study of genes, and you can have genetic markers and genetic disease, genetic modifications that have occurred in cancer, for example, or one's proneness to cardiovascular disease. And you have gene therapy. You can actually correct those. They call it the CRISPR method, CRISPR method of gene modification. And some diseases have actually been corrected through this method, where genes of affected people have been corrected. That's one. Metabolomics is basically a new term, relatively new term, which applies to metabolic products of both physiology and pathology. And it helps you in early diagnosis. So early, if you look at some protein that is produced by a cancer, for example, and if you can pick it up early, you have an early diagnosis, even before the cancer becomes detectable at the level of our scans, for example. Generally said that any tumor must be about a centimeter big before you can really detect it on our scans. And so much before that, as it is developing, you can diagnose it. Similarly, you can target knowing the metabolites that are produced and the pathways by which they are produced. You can block them by using antibodies, monoclonal antibodies, and various other things which are also in some form, it comes under proteomics. And very important is that all this involves huge amounts of data. And so you need artificial intelligence and bioinformatics. That's a huge topic by itself, a specialization by itself to, among computer professionals. Bioinformatics, large data, analysis, data mining, and building computational models. What was done in the animals, in the laboratories on animals, and then bringing it to, patient studies in phase 3 trials and things. All this is now shortened by building reliable and reproducible computational models, which tell you that if you put this chemical or this drug here, this is going to be the outcome. And that has really gone, advanced very much. And the other thing is this Internet of Things. You must have heard about Internet of Things, which has public health implications, where you have sensors, which are very reliable, and you can use sensors on large populations, collect data, and have policies which will help prevent diseases. So these are the main things. And I think we really should spend an hour discussing each aspect of this translational medicine sometime. Thank you.
I think the other thing, which I think sir is the latest thing is, for example, the advances that we have in terms of fish and, the hybridization and all these things. In addition, now what they are doing is, as sir said, instead of waiting for the tumor to actually appear on our investigative modalities, they are now able to count the number of tumor cells circulating in the bloodstream, and then they are able to then deal with them before they actually log themselves into some place and start multiplying. So even I think medicine has come to that level now, so that you are able to in nanosphere, you are able to count the number of cells and destroy them before they actually cause any problem. So I think to a large extent it is very exciting, and some extent it is scary as to where it is all going. And the other obvious thing is the cost implications. I think like most things, initially majority of these will be extremely expensive, not afforded by many patients. But as the advances happen, and as it is deployed in many people, obviously the cost will come down, and hopefully many people can afford it. I think it's very exciting, and I think we should have another class, I think, which will be much more detailed. This one, I think maybe, I don't know whether the students are willing to do it, or should one of the faculty give a lecture on this thing. So
we'll make it the group discussion. That way people read up about it, and we can then clarify doubts and things like that. Group discussion would be the way to go.
I am sure you'll agree. I think most of the students had come, read to some extent.
Yes.
Because everybody contributed to the thing, which was good.
Very nice. Yes.
Sir, Shrikant sir is also there, sir.
Shrikant sir, ready, sir.
Shrikant sir, ready, sir.
It is looking like Sanskrit. Some faculty should give a talk first, Sarita. Who is expert in this?
Who is expert? Nobody is an expert. We've all been just reading.
That's what.
We're all just
who is doing research on this? Some Indian Institute of Science or somewhere.
External
Author of the
faculty.
error.
That's a good idea,
Ah,
actually. Yeah, we should
that's
try and get
actually a very
somebody.
good idea. Correct.
Yeah.
If we invite, we'll find somebody. I think sir
Ah.
can,
Yeah.
we'll find somebody.
Are they doing any research in India or in Bangalore on this?
Plenty. You know, Kane Sridhar, my close friend, is an expert on tissue engineering.
Oh.
Yeah, his lab does only tissue engineering. We can really take a very, a very informed and, very detailed talk on tissue engineering. But I think, to me, it's good to know about tissue engineering, but I think translational medicine is a lot more exciting because it has a lot of
This current treatments have
applications.
become outdated.
Yeah, a lot of issues where tumors don't need surgery. That's what's going to happen in the next 1 or 2 decades.
Cardiothoracics.
Yeah. So I think Srikanth, any more comments?
Yeah, good. I think still more elaborate should have been. I'll find out if anyone can give a talk. I will.
Sure.
Yeah.
Please do. Let us know. Yeah. Okay, final remarks, Ravi, and then we'll
Niranjan?
close. Yeah. Niranjan spoke, yeah.
Yeah. Sir, I think with your permission, I'll just.
Sir, I think it was a nice novel class. I think. Yeah. So good presentation, Nikitha.
Thank you. So, sir, with your permission,
Yeah. Please.
I just like to, all of you to, today's this one, the meeting that you had all attended. I think sir made the point very subtly, but I'll make it much more, in a much more this one. Basically, when somebody comes with a mass abdomen in that area, I think, investigation, in a stepwise fashion before you consider anything like diagnostic laparoscopy or a surgery is very important. We should have considered ultrasound or CT guided biopsy. Even a CT should have been done, and colonoscopy and colonoscopic biopsy. And after that, then more invasive things like a laparoscopic, diagnostic laparoscopy and biopsy, or then whatever. I think sir made the point very well that despite all of that, if the histology came back as that, would you just leave it alone? But I think because this was not, it was very short-lived, we could potentially would have considered, watching this, kid for 2 or 3 months, and because if they are self-limiting, majority would have probably come down and maybe would have avoided a right hemicolectomy. So I think that is the lesson for all of us. Good night.
Yeah. All right. Good night. I'll close the meeting.